Hello guys, and welcome to Senses Lecture 4. We'll be covering eye structure as well as the basics of how eyesight works. So let's get started. First slide here is just showing you some of the muscles that move the eye, controlled by some of your cranial nerves like the trochlear nerve. If you can picture the muscle shortening, it pretty much tells you what it's going to do to the eye. If you take a look here at the superior rectus and imagine this muscle getting shorter, and you can see that it would rotate the eye upward. So you're going to rotate the eye up, you're going to rotate it down, you can turn it laterally, medially. This little chart here shows you all of those, as well as the cranial nerve that is controlling them, like the famous oculomotor, which pretty much says what it does. So know these muscles along with what they are actually doing. You don't have to pay attention to the cranial nerve function over here. As far as structure of the eyeball, you've got three major layers: a fibrous layer, a vascular layer, and then the sensory layer where you're going to find the rods and actual photoreceptor cells. If you're looking at these three layers, some of them are filled with fluids. There's fluids in between. You'll have a vitreous humor and aqueous humor. You also have the famous lens setting in there. We'll see all those in pictures here in just a little bit. Your fibrous layer is made up of two major regions, the sclera and the cornea. The sclera is protective in nature, mostly connective tissue, and that's where those eye muscles we just looked at are actually anchored to. The cornea is our transparent layer. It bends light, just like any lens basically does, right before the light actually hits the lens. Lots of pain receptors going through here, so this is why you tear up and have a feeling, the pain receptors from the eye. The vascular layer has got three regions: the choroid, a ciliary body, and the iris. The choroid is a region that supplies blood to the eye, so this is a nourishing region. The pigment actually helps absorb light. Ciliary body has ciliary muscles and the zonial suspensory ligaments, and it's going to function in controlling the shape of the lens. You can either flex the lens, making it, flatten out, or you can let it relax and get a little thicker. So you can control the shape of the lens using the ciliary body. If we take a look at this picture, we can see some of these structures we've been mentioning, like the cornea here. You have this little chamber filled with aqueous humor. Now, aqueous humor is actually produced down here from the ciliary process, and it'll flow up through this posterior chamber into this anterior chamber where you would find it, and then get reabsorbed down here. There's our suspensory ligaments, and you can picture this contracting and relaxing and changing the shape of this lens. The iris, this would be the pigmented part that we see that's green or blue or brown or whatever color your eye may be. The iris itself does absorb some light, and the opening is the pupil. This is where the light's going to pass through and actually hit the lens. So it controls how much light is going through. Your pupils can dilate in dim light and can contract in light that's actually too bright. Sensory layer, or retina, is made up of the more delicate layers. You have a pigmented layer, outer layer, that's going to absorb light and store vitamin A. A lot of your, pigments we're going to talk about that are sensitive to light that actually start the process of eyesight are synthesized from vitamin A. That's why vitamin A is so important for eyesight, like the retinols. The neural layers, where we find our photoreceptors, and these are going to be the rods and cones that are actually going to send the signal for light. It's going to travel down a bundle of nerves and out the optic nerve to the brain where it's going to be interpreted. But the process of getting the signal started is pretty complex. The cells that are going to transmit the electrical currents, like the ganglion cells and bipolar cells, are connected in with the photoreceptor. The ganglion cells will have long extensions like any neuron called axons. These axons kind of form big groups and form what's going to be called the optic nerve, which is going to exit out the back of the eye. And in that area there's no photoreceptors, so this is actually a blind spot. If we take a look at an image of what we just talked about, you have your photoreceptors here, rods and cones. So this is where light is going to change some chemicals inside these cells, which is ultimately going to start an action potential down here in these ganglion cells. And these are the axons that are running away. This is going to form ultimately the optic nerve. as it moves through. You can see this bundle here of axons. You can kind of see the image here we were just looking at. And they're going to exit right here, but you notice there's no photoreceptors at all in this area, so this is an actual blind spot for your eye. Another thing you might notice about the eye is for light to come through, it has to pass through quite a few cells before it actually hits the photoreceptors. It's kind of flip-flop. Rods, you want to know the basic difference between rods and cones? Rods are more for dim light. These are not the color vision guys. So these help you see in dim light, better suited for night vision. You get gray tones and things like that out of them, but this is not the color vision. Color vision belongs to the cones, but they don't activate unless the light is pretty intense. So you got to have pretty bright light for cones to activate. And we will get color vision from these, 3 pigments that we're actually going to use. If we're looking at our cones, you've got green, red, and blue. These are the wavelengths and their maximum absorption rates, so we can see along these color spectrums. And any combination of a reflection of green and blue and red can give you basically any color humans can see. This is visible light spectrum, so we live in this tiny little area. Many snakes have infrared vision, heat sensors. You can see up into here, kind of like those forward-looking infrared radars you see on the cop shows where the helicopter's chasing the guy around and you can see them at night. UV light, many birds see in the UV light spectrum. You know, we know what X-rays are. Most of us probably have had an X-ray or two. Gamma rays, often used in treating of cancer. Microwaves are pretty famous as well. And radio waves as well. So all these wavelengths of the electromagnetic spectrum we can't actually see. So what we see is very limited compared to what's really out there. If you're looking at activation of seeing long distance or short distance, a sympathetic nerve is going to relax the ciliary muscle here, which will tighten this zonule and these ligaments and flatten out the lens, which will allow you to see more long distance, something you're looking at that's kind of far away. On the opposite side of that, a parasympathetic impulse to this area will contract the ciliary muscles and relax these ligaments and make the lens kind of bulge. So this is for near vision, accommodating for something you're looking at that's really close to you. It's the movement of this lens that helps you accommodate for those things and allows you to see far and near. This is normal, perfect vision. You know, the light comes through the cornea and the aqueous humor here and then is bent by the lens and focuses back here on the fovea, and you see perfectly. This would be 20/20 vision, let's say, and normal. In a person who has an eyeball that is slightly longer than it should be, this person would be nearsighted. You focus the light, the focal point hits before it actually gets to the back of the eye, causing nearsighted. You can correct this with a concave lens. The opposite of that, if the eyeball is too short, is farsighted, and you focus, the focal point would be actually behind where it should be, so you get the fuzzy vision and you don't see so well. And you can correct that though with putting a convex lens in front of your lens and bending the light to where it fits perfectly. You can do this with contact lenses or glasses, either way. The chemistry of vision is where it gets a little more complicated. So this is how you actually see. It's basically a chemical change that's going to turn into an action potential, electrical current, that's then going to travel to your brain for you to interpret. The molecules, they're going to absorb the light and change. The retinols are combined with proteins called opsins, synthesized all from vitamin A. The two isomers we're going to talk about, 11-cis-retinol, the bent form, and all-trans-retinol as a straight form. When you convert the bent to the straight, that will initiate a chain reaction that's ultimately going to lead to an action potential. We're taking a look at one of our rhodopsin pigments here that's going to actually absorb the light. You can see the retinol here and the opsin proteins. When light comes in and in response to photons of light hitting it, this little protein, this 11-cis-retinol, changes shape, or this chemical, I should say. It activates a G protein called transducin. You can see the change from the bent form to the all trans form. Transduction activates phosphodiesterase, which will take cyclic GMP and convert it into GMP. This is guanine monophosphate. What that does is close these cyclic GMP gated channels. If there's no cyclic GMP, you're converting it all into just regular old GMP, then the protein closes, and this results in hyperpolarization. These ions are no longer free to flow in the cell, so the cell hyperpolarizes. And because of that hyperpolarization is going to start off our entire reaction that is going to ultimately lead to the action potential that causes us to see. So this covers the basics here of how the hyperpolarization is going to stop cells from releasing the neurotransmitter glutamate, and bipolar cells are no longer inhibited by this inhibitory neurotransmitter that allows them to depolarize, releasing their neurotransmitter on ganglion cells. Now the ganglion cells can start their action potentials, be carried down the optic nerve to the brain. Well, this will look a little more clear with pictures here in just a second. So in the dark, here's basically what's happening. You've got your cyclic GMP gated channels we've just seen that are allowing the cations to flow in and out, like they're supposed to. Basically what that's doing is you're not in a hyperpolarization state, you're depolarizing, and the voltage gated calcium channels are open, and neurotransmitter gets released continuously here. This neurotransmitter is inhibitory, okay, and it causes hyperpolarization in these cells, which means this cell here, this bipolar cell, is basically turned off. That closes these little tiny calcium channels that are on the axon terminal down here. So they're shut off because of the release of glutamate, the inhibitory neurotransmitter. So nothing's happening, meaning my ganglion cell is not getting a neurotransmitter signal from the bipolar cell. So because of the inhibitory neurotransmitter, because the cyclic GMP gated channels are open. Now when light comes in and strikes this photoreceptor and converts that 11 cis into the all trans, that's when you're going to start the transduction. The phosphodiesterase is going to convert this cyclic GMP into GMP, and this is going to change the system completely. So in this case, the cyclic GMP gated channels get closed because of the activation of the phosphodiesterase, of the light exciting the little photon receptors here. So the cation influx stops, and you get hyperpolarization. What that basically causes is no neurotransmitter release here. The voltage-gated calcium channels are closed, no calcium flows in here, so no neurotransmitter release from the photoreceptor. If this photoreceptor is not releasing the glutamate, the inhibitory neurotransmitter, then this bipolar cell now is basically turned on. So the lack of the inhibitory neurotransmitter turns on this cell. Calcium channels are now open. Calcium can flow in this axon terminal, causing the release of the neurotransmitter across the gap here. And this is an excitatory neurotransmitter. This will signal the ganglion cell to turn on. Now when this one turns on, the typical action potential, sodium in, potassium out, will propagate down the nerve, and that optic nerve will send that signal to the brain, back to the occipital lobe for interpretation. And you will be having vision at this point. Without light, there is no vision. Pretty simple. This covers the basics, a simplified view of it, that your photons of light are going to go through your cornea, then the aqueous humor, then through your pupil, then the vitreous humor, and then strike those photoreceptors we were just looking at, causing that somewhat complicated chemical change. So we just said of the retina causing a chemical change that starts an action potential. That's what's happening without the details of the glutamate and the 11 cis retinol converting into all trans and all the things we just discussed. This is going to travel down the optic nerve to the brain, occipital lobe mainly, for interpretation. And that's how vision basically works.